Good morning. This is my first time doing a live presentation, in-person presentation with a mask on. So if I sound muffled, like raise your hand and I'll repeat what I said. So just what I'm going to present here is just some background on work that I've been doing with the Center for Behavioral and Addiction Medicine at UCLA in collaboration with the Department of Epidemiology at UCLA too. And so it's not, oh, there you go. And so when I came on board at UCLA, my interest was in understanding initially the relationship between social adversity and the intersection of methamphetamine use and HIV comorbidities. And so I came to realize that one thing that was missing in a lot of this formula, in this understanding this relationship, was some of the biological mechanisms that were at play here. And so a lot of times when we think about stress, we tend to talk about it as like something that's just kind of in our head, something that we experience, but there's a whole biological process that's going on underneath all that, which I want to talk about today. So just as one thing that we've been doing is that we're trying to understand, like, some of the conversation has moved away from just like sustained methamphetamine abstinence to the reduction in methamphetamine, so that we understand that recovery is a process and that there is a spectrum of response to methamphetamine treatment. So here, I just want to present some epidemiological results here. So what we kind of wanted to understand was when we look at different levels of methamphetamine use, so we're looking at none versus monthly or less or weekly or more. This is kind of the best way we can understand dosage or dosing. And so with escalating levels of methamphetamine use, we actually see that level of use seems to correspond with escalating risk of some of these other different conditions. So the first things that we looked at was adverse social experiences. So this includes unemployment, housing instability, intimate partner violence. We've also, in a prior study, showed that experience of homophobic victimization was also linked to methamphetamine use at escalating levels. We also looked at sexual risk behavior. So this includes like having anal intercourse partners, multiple sex partners, exchange sex, and then escalating risk of positive STIs, definitely increased risk of viral load in people living with HIV. And then we also have these clinical comorbid conditions, including a neurological condition, renal condition, and psychological conditions. So this is the one, I'm going to run through this real fast because I've already talked about the experience of homophobic victimization and unstable housing. This was shown in the longitudinal analysis where we actually lagged the experience of homophobia, meaning that we were able to say that prior experience of homophobia was linked to subsequent housing instability and methamphetamine use. And then here, this is an evaluation we did of the LA County Medical Care Coordination Program, where we're trying to understand whether, then this is for people living with HIV who have complex need, and that stimulant use, especially when comorbid with other psychosocial comorbidities, including depressive symptomatology and housing instability, had the worst response to treatment. So, I mean, by worst response, I mean they still responded to the treatment, but they didn't achieve the same probability of viral suppression as everyone else. So, the area of biology is kind of more like understanding the transcriptome, basically, and how this ties together methamphetamine use, adverse social experiences, and clinical health outcomes. So when I talk about gene regulatory pathways, I'm not talking about trait genes, so I'm not talking about trait genetic differences, but I'm talking about more just on a higher level, it's just gene behavior. So how do our genes respond to methamphetamine use, to stress, and to HIV? All right, so the field of social genomics has kind of created this theoretical model, which has been tested, called, they call it the conserved transcriptional response to adversity. And there's this kind of signal transduction that happens. And I'm not going to take you through the nitty-gritty of the molecular stages and stuff like that. But basically, just in summary, through multiple studies, including one that I've done on experience of homophobia, and some research that my colleague Steve Cole, a senior mentor of mine, has found that experiences of racism and experiences of homophobia, poverty, loneliness, housing instability, family conflict are consistently linked to inflammatory gene expression, and type 1 interferon activity. So in people living without HIV, people who are not using substances, the mechanism is a little bit more simple. Type 1 interferon activity, which is our viral response, actually seems to go down, whereas inflammation seems to go up, typically. Again, that's in the absence of methamphetamine use, absence of HIV. So, when we think about social adversity, so that was, kind of the basis for understanding social genomic processes. We understand that when we think about substance use, we can't think about it in the absence of infectious disease and adverse social experiences. So it's important to look at kind of these things together, especially when we're looking at multiple minority groups and those with who may have comorbid stimulant use, HIV, and social adversity. So all these conditions can dysregulate the immune system. So the purpose of the study that I recently did was to look at how methamphetamine use, unsuppressed viral load, and multiple domains of social adversity are linked to changes in inflammatory and antiviral gene expression. The thing that's interesting is that for those of you who have been keeping up with the COVID-19 research and how interferon response plays a big role in the response to COVID-19, those with an inadequate antiviral response or interferon response are the ones who tend to have the cytokine storms. When we think about this conversely with HIV, when we have a heightened interferon response, that can lead to actually, things like T-cell exhaustion and viral replication too. So it's kind of the inverse when we're thinking about the context of HIV. So this gets a little bit more complicated. So if you're kind of wondering, like, is innate antiviral response high or low? Is it good? And it's like, kind of depends on what the context is. So anyway, anybody have a lot? Is everybody still with me for now? Okay, cool. All right.
I had a quick question about some of the earlier slides. I mean, what's your sense of the causality there? Like, what's the direction? You can imagine that adverse social circumstances could lead to increased meth use, but you could also imagine that increased meth use leads to adverse social circumstances. So is there any work on, causal inference for?
Yeah, so this is with like a longitudinal cohort study. So ascertaining direction of causality is a little bit difficult. So just to let you know, everybody in the room is nodding with you. So yeah,
that's unusual.
So this was a way
for this moment.
Yeah, like for this slide right here, we tried to kind of get at that where we're looking at prior use, we're kind of controlling for baseline and then following them up over time. And so there seems to be, I've been able in just like exploratory analysis to confirm reverse association. And then also where we have social adversity preceding methamphetamine use and some of the other social adversities. So, there was work with the Williams Institute showing that people who experience homophobia in the family were more likely to be unstably housed later on in life. And, reported that was the reason why they left their home. There's research, of course, indicating that methamphetamine use, of course, can lead to things like unstable housing because it causes disorganization in a person's life. Suggests that I think it's, for now, what we have right here is more associational right now, but there are methods that we've been, in this kind of next stage of research trying to ascertain directionality. So thank you for that.
So right now it's unknown. I guess that's helpful.
Yeah, well, so here's this with the county, for example. So this is actually baseline substance use for people who were entered into this program. So people who started with substance use, upon entering the program, they actually had less subsequent response to this comprehensive HIV care program offered by the LA County. So in this case, we see stimulant use interfering on some level, whether it's through a social mechanism or through a psychological mechanism, behavioral, or sorry.
Sorry, and how do you measure success?
For this program, so we use.
I'm sorry, so we can't hear people in the room. So if you would just repeat.
So his question was like, so what is the outcome that we're looking at right here with this slide? And so viral suppression we defined as like less than 200 counts per milliliter of blood for a virus. And so the probability of viral suppression here, so if somebody ends up virally suppressed and we actually follow them throughout time, if they stayed that way, it looked like for the most part, once people, after about 6 months, people kind of stayed steady as a group. But however, right here, like 65% response to a program is still not quite where we want to be. We want to be much, much higher than that with viral suppression. So in this case, we are actually able to ascertain directionality of substance use preceding viral suppression. Does that make sense in this case? So in some cases, we've been able to show both directions.
Yeah, great, thank you.
Yeah, you're very welcome. Thanks for the question. All right, so just to let you know, what I work with, I don't do a lot of exploratory or like kind of empirical analysis of immune genes. I kind of use like an a priori approach based on what's been validated in prior transcriptome research. And so there's about 53 genes that seem to track with experienced social adversity. So I have this in this, I'll be sharing the slides later on with Cherie if people want to take a look at this. But this figure right here, we see that these are the inflammatory genes that seem to be consistently expressed as a result to experience social adversity. And the downregulated gene, type 1 interferon genes that also respond to social adversity. Now, again, remember what I said was that with HIV and with methamphetamine, things can actually go in the different direction because that complicates immune processes separate from just social experiences. I'm not gonna, so our main predictors right here, we're looking at these 3 different domains of social adversity: housing instability, intimate partner violence, homophobic victimization. These are comorbid with methamphetamine use. We're also controlling for unsuppressed viral load. So that's, by unsuppressed, that means greater than or equal to 200 count. And then methamphetamine positive urine test. And this is from the M-study cohort, which is here at UCLA, and it's a platform for longitudinal research that anybody can submit concepts to. So just what we found was that experience of methamphetamine use was positively associated at the bivariate level with, with inflammatory gene expression, and still was intimate partner violence too. So that's consistent with the literature on methamphetamine inducing peripheral inflammation. These are mRNA from leukocytes. And with intimate partner violence, we've seen that, in prior social genomics research, that social conflict also increases inflammation too. And so also with regard to interferon expression in people living with HIV, the type 1 interferon response went up as well. So normally with social genomics, again, remember I said that antiviral response goes down. With methamphetamine use, it goes up. Same thing that we're seeing with unsuppressed viral load. This makes sense too, because when you have unsuppressed viral load, you have increased monocyte activation and all that kind of stuff like that, and then increased T cell activation, and then the interferon response actually further drives viral replication. Interestingly, so this is what definitely seemed to go in the opposite direction, was experience of intimate partner violence and housing instability both drove up the antiviral response in the context of meth use and HIV. So, and this is with adjusted analysis. This is where we see percent change or percent increase right here. So these are confidence intervals with point estimates right here. So just so you can see it more visually instead of just looking at parameter estimates.
Could I ask a quick question?
Yeah.
So this is Roham. So, as people who are not experts in the field, one of the things that we've always wondered is whether the route of amphetamine use is also associated with differential inflammation. If I snort it, is it the same thing as if I inject it or ingest it? Is there data? I know this is a little bit different than what you're presenting here, but it could also confound this. Does the route of use also influence these patterns?
I think I would keep that question in mind for when we start getting into compartments that get affected by methamphetamine use, because I can see that coming into play. For example, we have Jen Fulcher in the room who works on the gut biome. So I imagine that, for example, like ingestion, for example, I don't know, versus like injection versus if we think about snorting or smoking, that probably has implications too for respiratory pathways. So I think since this is white blood cells, we don't get into other compartments, but I think that is definitely an area for research. And I think that's probably the next direction that we're going to be going. Yes. So Steve Shoptaw was saying that as far as addiction, as substance use disorder, like addiction, like there's not a difference there. But when we think about chronic disease, I don't, or you know, pathogenesis and molecular, I don't really know. Oh, and just to let you know, we're going to bring the mic over to another expert right now who's going to chime in.
So there's one study from Adam Carrico's group, and I helped them with this, where they did look at plasma inflammation markers in people who injected meth compared to people who did not inject meth. And there were increased IL-6, soluble CD163, and I think LPS binding protein.
Okay.
Those who are injectors compared to those who are not.
Okay.
That's the only data that I'm aware of.
Okay.
The route of use.
What were the markers again? IL-6 and what else?
IL-6, soluble CD163, and
okay.
LPS binding protein.
Okay, soluble CD63, you said, right?
63.
Okay.
Yeah.
Thank you.
And those were independent of other characteristics, because I imagine there are certain personal characteristics of injectors versus non-injectors.
Yeah, I can't remember off the top of my head all of the covariates that were accounted for, but they definitely did control for as much as they could.
Great.
Yeah, just to let you know, so that was plasma biomarker. So this, kind of like you were saying, this is a little bit different because this is looking at within cell changes versus like plasma, which is like, you know, outside of the cells. So it's, yeah, those are a very interesting question. I think that's where the research is at so far that we know of. Any other questions before I jump forward? How am I in time? Okay, cool. So again, so this also confirms our findings. These are just adjusted analysis of what I presented to you before was just bivariate analyses. So still confirming when we look at all of these three things together, methamphetamine use, unsuppressed viral and intimate partner violence, you know, net of effects of each other, we're still seem to be driving up type 1 interferon expression. So that kind of summarized our findings. So basically, the implications of this is that when we look at social genomics research, I think it's important when, you know, I think a lot of times we think of social adversity as happening in a vacuum in the prior research, but I don't think that we can really think about it that way anymore now that we understanding that substance use and HIV play a role in people's social experience and also in chronic disease pathogenesis. So when we look at this, you know, dysregulated type 1 interferon and inflammatory gene expression, you know, it's important to look at these things through kind of a comorbidity lens. And so the prior social genomics research has, you know, linked this dysregulation of inflammation and type 1 interferon expression to, you know, psychiatric disorders, anxiety-based disorders, and depression, cardiovascular disease, metabolic disorders, and even some cancers longitudinally. And this is kind of the next stage of research, is to kind of follow people through the life trajectory and follow, you know, and understand the stress biology mechanism at play here. All right, let's see. And then also for people living with HIV, if there is methamphetamine use at play, you know, elevated inflammation and also elevated type 1 interferon expression are important markers that we look at when we're trying to understand T cell exhaustion and HIV viral replication. So the next stage of research, so some of this is kind of like you were saying, was like understanding directionality. So longitudinal experimental studies are needed to, for one thing, just test the validity of these markers as, you know, kind of like a, you know, clinical monitoring tool alongside others, you know, standard clinical endpoints. And then and then looking at the utility of these social genomic markers as indicators for disease risk is also could be useful for understanding mechanism. And then potentially using it as a tool to understand a treatment response to in the context of comprehensive care. So that's all I have here. This is the, you know, myriad studies that have research grants that have been supporting this collective work right here. So I'd like to acknowledge these, including the M study, my K award, which I've coined Express Plus, the T32 postdoc that I was in for a couple of years, and the center grant, the UCLA USC Center on Biodemography and Population Health, which is supporting the Social Genomics Laboratory. I'd also like to acknowledge the Center for HIV Identification, Prevention, and Treatment Services too, and the UCLA CTSI. Any questions? Yeah. Yes, I can hear you.
Question is, the social genomics associated with inflammatory response and associated with chronic disease seems to
Mm-hmm.
how do you think about it in terms of its uniqueness for methyl versus depression? How do you want to start thinking about it so that ultimately we can make it point in time, but we have to start thinking about how to do it. Any signal that we have to
This might be even like a little too far reaching, but one of the things that Steve Cole has been talking about with this line of work is interested in using these markers as a form of biofeedback for participants in treatment. So if it's in the context of depression treatment or methamphetamine use, one thing that might be interesting is for people to actually like look at this as a marker of change, because sometimes like you might feel improvement, but actually showing like a marker of improvement, like, oh, you know, we've been running these blood tests on you and these things seem to be going back to, you know, a form of homeostasis or kind of returning to, you know, reduced levels or that sort of thing. So that is one approach in terms of a implementation approach. I'm not sure if I'm answering your question quite. Now,
I'm just
have you been able to at least begin to discern what may be different pathways for for the inflammatory response that then is associated with meth use as opposed to, say, depression? Or is it just a common sort of global risk factor for one of these and then the disease happens because of other things? How do you think about
Yeah, so what I did was I used the a priori genes from the standard social genomic, you know, kind of scaled or contrast that they were using. So to your point, that there might actually be other expressed genes that are different from what was in this a priori gene set. So that would be the next stage of the research. We'll be doing some empirical exploratory analyses on these, see how this deviates and maybe ways it adds or contributes to some of these other like the more psychological comorbidities and stuff like that, and the ways they intersect. So that would be the next stage of research right now. So this was just a priori, and the next stage is like, how do we refine this and or even like expand it? So I think I would say just because we're seeing, for example, the interferon response is going the complete opposite direction, to me, that's already a sign that we probably need to look at this with like openness to maybe some other markers that need to be maybe contributing to this scoring. Yeah.
Just a quick question. I thought this is really well presented, and I may not be thinking of this right, but are there any genetic risk factors of meth use or enzymes that could be used like in a Mendelian randomization to get at whether, you know, use of these things impacts on these inflammatory markers in HIV? Is that a sensible question? I'm not sure.
I think that's a fair question. I'm not as familiar too much with trait genes. So one thing I am like a little bit I followed, for example, was the mutation to DRD2 receptors, for example, dopamine, and I'm not sure how that might end up playing into, you know, maybe a response to some of these inflammatory processes. But it does seem to indicate that, you know, there's more challenges with response to actual methamphetamine treatment when we see that kind of genetic difference here. So what I've been more in the realm of has been like kind of gene behavior and less about trait genes, but I think that's a very fair question, and there might be some people in the room who are better suited to answer that question later. Did that kind of answer your question? Okay. Anybody else? All right. Well, thank you for your enthusiasm and for taking this conversation to exciting places. So.